Thank you very much, Rob, first of all, for this very kind introduction. And good afternoon, everyone. Indeed, I'm Alessia Candeo, and today's talk will be about a multimodal and multiscalar hyperspectral imaging technique, and I will also showcase some of the results that have been gained with this method. I will first of all start with introducing myself another time with a picture of myself. That's me. I'm on the rooftop of our physics department. That's not actually just a picture of me, but it's an hyperspectral image. So it's an image where we have for each pixel not only a spatial information, but also a spectral information. In fact, if we select single pixel or single ROIs or region of interest, we can retrieve a spectral information that can be a transmission, a reflectance, a fluorescence information about the material located at that specific location in the picture. This is particularly useful because it allows to study materials with also a spatial information. And spectral imaging or hyperspectral imaging has been applied to multiple scales, from the microscopic one to the large scale one. And it has been used on many different subject areas, running from biology, nanostructures, food, cultural heritage, earth monitoring, and let's say improvement of materials. In general, let's say that there is a very basic way to obtain a spectral image or an hyperspectral image. We just illuminate our object of the interest, we image it with an imaging system, and then we send the retrieved image to a wheel with filter, so a rotating filter wheel, for example. And in this manner, we can acquire many different images with a different spectral content. In this way, we can also retrieve a so-called spectral cube in a pretty efficient manner with this wide field technique. But of course, in this way, the spectral resolution is low because we are limited by the bandwidth of the filters that are being used. Another way of obtaining an hyperspectral cube is called line scanning or push broom imaging, in which we have again an illumination source. We illuminate our object, and then with the line filter, we select only single lines of the object, and we send them through an imaging system and then to a dispersive element, for example, a spectrometer or a prism. And then we collect the spectral information of single lines. So to retrieve an image, we have to scan the line filter throughout the object. This technique has a higher spectral resolution. We're talking about 2.5 nanometers at 600 nanometer wavelength. But the problem here is that the light throughput is quite low because we have a spatial filter, the line filter, in fact. And also it is a scanning method. So it is quite useful. Of course, it's very useful, but mainly for flat and small samples. Another approach, a completely different one, is the Fourier Transform spectroscopy-based method, or the interferometric method, in which basically we generate two replicas of our incoming light, and we separate them in time with specific delay. And by varying this delay, we can basically let the two replicas interfere. So with the photodetector, we can acquire different signals, let's say, of this interference between the two replicas, and we can obtain a so-called interferogram. And then with the Fourier Transform method, we can retrieve the spectrum. Of course, if instead of the photodetector, we use a camera, we can also retrieve many different images at different delays, and then obtain a so-called hyperspectral image or a hyperspectral cube through a Fourier Transform calculation. The most common way to perform a Fourier Transform imaging or to obtain an interferometer is to use a Michelson interferometer, where the incoming light is split into two replicas through a beam splitter, and then the two replicas are sent to two different mirrors, and by moving one of the two mirrors, we basically can tune the delay between the two replicas. And then the interferogram can be acquired either with a photodiode or with a camera. And it is indeed possible to obtain the spectral information. This method has a high light throughput, and it is a parallel acquisition. So we have a wide field hyperspectral imaging that we can obtain. And also it's quite flexible because it allows us to tune the spectral resolution by acquiring long or short interferograms. However, there is also a drawback because these interferometers are quite unstable. They are cumbersome to implement. And this is because they are, in fact, they are used for the measurements of vibrations. So it's quite intrinsic that they are unstable. Our method is similar but different. The generation of the two replicas is based on the use of a birefringent crystal. And in our case, we have a common path interferometer where the incoming light basically can travel, can be split into two replicas, one running on the ordinary axis of the birefringent material and the other one running on the extraordinary axis of the same. And when they exit the crystal, basically they have a delay between themselves. And of course, if we could tune the length of this crystal, we could change the delay between the two replicas. And in fact, that's exactly what we do, because our birefringent crystals are shaped in the form of wedges. And by moving one wedge with respect to the other, we can change the overall thickness of the birefringent material and tune the delay between the two replicas that then are put together again through a polarizer. And indeed, we can acquire again an interferogram, and then a spectrum can be retrieved. This ultra-stable common path interferometer, which we call TWINS, or translating wedge-based identical pulses encoding system, has been patented at the Department of Physics here. And it has a very good spectral resolution with 4 nanometers resolution at 600 nanometers, so comparable to the one of the push room approach. It is virtually alignment free. It is very robust because once you have aligned it, it stays aligned. So there is no stability control required, and it is insensitive to any mechanical fluctuation. What's more, it's also compact and it can be mounted on any system, also thanks to the fact that it's very lightweight, and it is portable, meaning that we can carry it around outside of the lab, on the plane, on the train, in a shoebox. Thanks to the high throughput of this system, I'm talking about the light throughput, it is also possible, for example, to use it in very low light conditions. And if we couple this interferometer to an imaging system, just by adding basically an objective and a camera behind the interferometer. And in this manner, we can you still hear me?
Yeah, we still got you, Alessia.
Okay, so and in this way, we can look at fields of view that can go from 30 centimeters to 3 meters with a spatial resolution of maximum few millimeters. However, by just adding a further macro objective in front of the interferometer itself, we can move to a macro imaging system with a field of view of 1 to 2 centimeters and a spatial resolution that is lower than 10 micrometers. Lastly, we can also couple it to standard microscopes, commercial ones. In this case, of course, the field of view is given by the objective lens being used, and the spatial resolution is preserved. These are the different implementations of our hyperspectral camera. You can see that we can use it for large field of view in a macro configuration and on a standard commercial system. What's more, we can also perform multimodal imaging, because with this system we can acquire both fluorescence, reflectance, Raman signals, even transmitted light, and we can combine them overall to improve the understanding of the samples being acquired. I would like to start focusing a bit more on the macro scale implementation of this hyperspectral camera. When we use our macro imaging system, it's because we want to maximize basically the signal collection over a smaller field of view. And for example, we've used this configuration of our interferometer for the study of biofilm formation on stone samples, for example, for conservation science, for statues that are placed outdoors. And in this case, we have observed a field of view of 15 by 15 millimeters with a resolution lower than 10 micrometers. We decided to acquire both reflectance and fluorescence dataset. We've used the reflectance dataset to correct for the self-reabsorption of the fluorescence dataset. And we've used an halogen lamp for the illumination of the reflectance dataset and an LED excitation for the fluorescence dataset. And the acquisition of both datasets took less than 5 minutes. If I now focus a little bit more on the results that we obtained on these cyanobacteria biofilms on limestones, I'd like to show you the data collected with the reflectivity during the reflectivity experiment. You are looking here at, on the left here, we have a standard photographic image of the limestone sample, and then we have here a false RGB image that we obtained from the reflectivity spectrum, the reflectivity data set. And you can see that there is not much to be seen with visible light, but then we can also select different areas on this image to extract these reflectance spectra. And thanks to the quality of our data, we can also compute the first derivative of the reflectance spectra and select, for example, the maxima of this first derivative of reflectance spectra and pull out the specific images at that wavelength. And we can color code them with blue, green, and red color to create a composite image where the red color, where the red channel is correspond to the first derivative at 680 nanometers, the green color correspond to the 655 nanometers first derivative peak, and the blue channel correspond to the 550 nanometer first derivative peak. And through this composite image, we can start saying that there is at least there are at least 2 biological materials that are present on the surface of this stone. With the fluorescence dataset, we tried to see if we can see something more. And with the false RGB, there is not much to be seen because the image looks very uneven. But then if we move to a false RGB of this fluorescence dataset, we can start seeing that there is actually a lot of biofilm present on this stone. Everything that is fluorescent is basically biofilm. And in fact, even if we select the gray area here, we can even say that even in areas where there appear to be nothing, there is actually a little bit of signal coming from biofilm of the cyanobacteria. So the technique is very sensitive even to traces of the biofilm. And then we can, from the fluorescence spectra, we can understand that there are 2 main peaks that we are observing, one at 700 nanometers, that is corresponding to the 2 photosystem PS1 and PS2 of the chlorophyll, of chlorophyll. And then we have another peak at 685 nanometers, which corresponds specifically to chlorophyll A. And then we can observe that this blue shift in the peak of chlorophyll A actually corresponds to degradation products. And so we can tell that in this biofilm, there are areas where the biofilm is healthier than others. If we look at the 710 nanometers peak, instead we can say something about the bioactivity of the biofilm, because this peak, the higher the peak, the higher the photosynthetic activity in this case. And then we can plot maps of the wavelength of the chlorophyll A peak, which basically tells us something on the biofilm degradation, or maps of the ratio between the emission peak at 710 nanometers and the emission peak at 685 nanometer. This map is basically telling us where are the areas where the biofilm is more photosynthetically active. And this area actually corresponds to fissures or cracks in the stone. Thanks to the quality of our datasets, it is also possible to perform clustering. Specifically, we can perform both unsupervised clustering with very standard methods like k-means, but also supervised methods, like, for example, spectral angle mapping. This is something we use a lot. Basically, we select spectra in the image or even from a library, and we look which are the other spectra in the image that are the closest. And based on that, we can create maps of similarity, and then we can also create composite maps of similarity, which let us know something about our sample. This method has been applied also, for example, to the study of dye degradation and fading of dyes in tinted fibers. Specifically, we looked at the fading of indigo carmine on wool fibers. And in this case, our collaborators provided us with aged samples that showed discoloration with aging. The technique looked to be very suitable for the early detection of the degradation of the dye, and also it was sensitive enough to detect very faint traces of the eye, even in highly discolored material. From the reflectance dataset, we can first of all observe that there is a very nice resolution with all the fibers in focus, and we can even select single strands of the thread of wool, and we can again retrieve the reflectivity, the reflectance spectra, and the first derivative spectra corresponded to the selected areas, and we can again map, for example, the peak of the first derivative to understand where are the areas where the pigment is more highly degraded. From the fluorescence, we managed to obtain information of traces of the dye that was under study, especially when we look at the false RGB images. And through those, we can also detect the presence of uneven fading on the very same fiber. This is something very hard to get with pointwise techniques like Raman microscopy or in general liquid chromatography that are usually being used for this type of studies. And also in this case, we can look at how the spectra changes with aging, and we can perform the computation of the map of the ratios between the emission, for example, of the wool in this case, and the dye, to observe where untouched dye is still present, or we can even run PCA analysis to study this distribution of the degraded sample, sample dye. The camera can be used also to perform large field of view imaging. Like in this case, we are looking at the Pusiano Lake in the north of Italy, where the camera was brought to. So the camera is very portable, it doesn't use a lot of power, so it can be easily used even in adverse conditions if you want. And it was used, in fact, for outdoor images, and it can be used for more in general for remote sensing. For example, here it was possible to see the differences in the chlorophyll of different trees on the lake. It was possible to, of course, reconstruct the color image. It is also possible to perform a study through with the transmitted light, so transmitted light hyperspectral imaging. For example, we studied this artistic glass window. We just went to the Church of Santo Spirito in Milano with the camera, and we focused our attention to this detail of the glass window. We retrieved our hyperspectral images of this part of this portion of the glass window, and we realized that all these different blue tiles were actually made of, they had different spectral characteristics, they were different types of glasses, and in fact we could segment them based on a k-means approach that allowed us to see that in fact all these tiles have different properties. It was possible to carry our instrument also, for example, to the CDRMF at the Palais du Louvre in Paris. We used the camera to, for example, study the different distribution of pigments on painting. And for example, in this case, it was possible to map all the different pigments that were used in this Indian folio. We use the camera also to map the efficacy of cleaning and conservation on this bas relief, for example, that was in fact monitored before and after cleaning. And it was possible even for such a large object to perform photoluminescence studies. In fact, fluorescence or photoluminescence is highly sensitive to the presence of organic contaminants, for example. And when we highlight, for example, this specific detail, it is quite obvious that there is presence of still some organic contaminants on top of the gypsum. And we also can detect the presence of the materials that were used during the conservation. Thanks to the high light throughput of the system, it is possible, as I previously mentioned, to perform experiments even on large field of view with fluorescence of highly three-dimensional objects. And this is due thanks to the fact that we can close the diaphragm of our objectives while still having a good signal to noise ratio. And in this case, we have studied this statue that was vandalized, and we focused our attention on this detail. And it was possible to understand in which areas there were organic pollutants or synthetic pigments that were used during the vandalism to guide the conservation process. Lastly, I'd like to mention that we also looked at the early detection of damage and pest invasion in plants. We have basically looked at what happens when you change the temperature locally on a leaf, on a full plant. And we observed a variation in the chlorophyll emission even immediately after the increase in temperature. So it was possible to detect this thermal damage very early. And it was also possible to study, for example, the early stress induced by pest invasion on crop leaves. Lastly, I will move to microscopy. As I mentioned, the system can be easily adapted for use with commercial microscope, like this Leica microscope we had around the lab. And with the microscope approach, of course, is possible to study biological sample. Here we were just looking at 2 fluorophores that were used for the staining of these Caco cells, but we could have many more overlapping dyes, for example, and perform spectral unmixing with this system. It was also possible to study the material distribution in stratigraphic samples. For example, here we had a microfragment that was taken from a violin and embedded in epoxy resin. And we looked at one of the cross sections, and it is quite clear already from a standard photography and UV illumination that there is a very, let's say, complex distribution of the materials on the surface of this violin. But with our camera, it was possible to cluster, let's say, the different types of materials based on their proteinaceous emissions or resinous emission. And it was also possible in orange to map alterations of the varnish that was used for this for the top layer of the violin. We use the system also for the characterization of new films for the food packaging industry. Specifically, in this case, our collaborators provided us with some samples of commercial food packaging that were coated with carbon dots to improve the rejection of UV light arriving to food. And it was possible to map the distribution of carbon dots and look at their aggregation. For example, in this case, PLA was covered with carbon nanodots with 2 different techniques, spinning or spraying, and it was indeed possible to evaluate the let's say, the extent of aggregation of the carbon materials. And indeed, this is a good guidance for the development of new types of commercial food packaging. Lastly, I would like to mention that we can also assess the quality of 2D semiconductor. For example, here we study molybdenum disulfide samples. We were provided with samples of monolayer and bilayers of MOS2. And already from the reflectance image, it is possible to observe that there is monolayer, there are monolayers and multilayers. Just by looking at the contrast of this reflectance image. But then when we looked with the fluorescence and our camera, it was quite obvious that there was, yes, a bilayer present, but also the monolayer was not very flat, well, actually was not very, let's say, homogeneous. It didn't have a good quality because the emission of the fluorescence was not even, let's say. And this can be due to strain induced, for example, electronic band gap variations, or in general fluctuations in the dielectric environment. Then we can also use our camera in all these 3 different configurations to perform time-resolved hyperspectral images. In this case, basically, our interferometer is coupled to an objective and then an intensifier and a camera that is a time-resolved or a time-gated camera, let's say. In this case, basically, we have to use a pulsed laser, and a pulse of laser excites the fluorescence from the sample, and then we can open selectively our gated camera at different delays with respect to this pulsed excitation. And in this manner, we can look at the emission at specific delays. This is particularly relevant when there is a variation in the spectral behavior of the materials at different temporal windows, and especially when we have long, let's say, long decays of the materials. This system, this time-resolved hyperspectral imaging system, was used to perform experiment on stratigraphic samples, for example, from this Allegoria in Memoria di Francesca de Maestri Colleoni, which is a painting. And in this case, we have a visible image and also a UV image of a standard photo taken with a standard photographic camera. We can tell that there is some emission from this sample, but we cannot really say much about the composition. While instead, if we perform experiment in the nanosecond time scale or in the microsecond time scale, it becomes more obvious what, for example, layer 1 is made of. And specifically, if we look at the nanosecond time scale, these are the spectra that were retrieved, we can see in blue that there is no much emission from this layer 1 between 400 and 900 nanometers. But then when we move to the microsecond time scale, we see that the layer 1 has a very clear emission peaked at 530 nanometer, which is the typical emission in the microsecond time range of zinc white. So while in the nanosecond time domain, we look at the zinc white band gap, which is in fact before 400 nanometers, in the microsecond time range, we are looking at zinc white trap states, and we could assign in this manner to layer 1 the main element that was present. We also performed time-resolved hyperspectral imaging on wide samples, like for example this painting from Aldorfer. And we have focused our attention in one specific region here, where the feet of Christ are positioned. In this case, we perform nanosecond, microsecond, and also millisecond time-resolved imaging. And you can maybe see that in the nanosecond time scale, there are areas that are dark, that are not emissive, or at least their emission is not visible because it's covered by all the other pigments emission. But when we go to the microsecond time scale, we can start to see a new emission, which is again emission from zinc white, peaked at 530 nanometers. Here you can see the spectrum. And if we go to the millisecond time scale, we can still see the tail of the emission from lead white, which has this specific spectrum highlighted here in red. And all this was confirmed with XRF mapping. Lastly, we have recently implemented our first wide field hyperspectral Raman imaging system, where basically we have used the microscopic configuration of the twins coupled with a 532 nanometers laser. As we might maybe all know, it's always hard to discriminate between fluorescence and Raman emission from samples, but with an interferometric approach, it becomes easier. In fact, a wide bandwidth signal like the fluorescence one has interferograms that are quite short, let's say, or at least they finish very, very early. Let's say that after few cycles, they are done, they are finished. The interferogram is very short. But instead, the Raman signal, which is very narrow, has a very long interferogram, okay? Because the signal, let's say, from the interferogram point of view, lasts for very long. So it's present even at very, very large delays. And so if we selectively acquire data and, like, let's say, if we selectively acquire short interferogram, we can favor the emission of the fluorescence, the retrieval of fluorescence, while if we perform long interferograms, we favor the acquisition of Raman data. And to have a good Raman resolution, it was important to have very long scans, very long interferometric scans. And to this purpose, we had to change our crystals from alpha BBOs to IVO crystals. So our wedges had to be changed. And this is due to the fact that basically IVO has a delta N that is larger. It has basically the difference between the ordinary and extraordinary index of refraction is bigger, and this allowed us to perform extremely long and also the apex angle of the wedges is larger, and this allowed us to perform very long scans of our interferometers and achieve resolutions of 25 centimeters to the -1. With this system, it was possible to discriminate, for example, between polystyrene and PMMA beats in a very easily manner, and we could retrieve both the fluorescence and the Raman spectra. The Raman instrument, the hyperspectral Raman instrument has been also used for the environmental pollutant identification and characterization. For example, in the scope of understanding how textiles lose microfibers. And in fact, it was possible to, of course, observe the presence of microfibers on an aluminum substrate and perform Raman and spectral angle mapping of the Raman results and distinguish between polyester and polyamide fibers. Finally, it was also possible to detect and identify microplastics on filter membranes. For example, in this case, we have membranes. These are membranes that are commercially used for filtering microplastics. They are made of cellulose, and it was possible to distinguish on cellulose the presence, for example, of polyethylene. I'd like to conclude this talk just by mentioning that we are now moving to the near IR, because in this, let's say, spectral window, textile materials, for example, have a high, they have a very specific spectra, and so this can be very useful, for example, in the field of sorting of waste textiles. So we are now, right now, we are implementing a new hyperspectral microscope that can acquire spectra from 400 nanometer to 1.7 micrometers. And with this, I'd like to conclude. Thank you for following this talk. But also thanks to all the people involved in this research. Most of them are in this hyperspectral image, but not all of them. But still, I thank everybody for working with me on this research.